So today, this is our 7th consortium meeting already. It all has started at the very same spot here in Tours at the Hotel Univers. Here, this is a picture of our kickoff meeting in 2017. Then we had the meeting in Weybridge in May 2018. After that, we had a meeting in Madrid in May 2019. Then there were the COVID years, during which we had just 2 online consortium meetings. And then again in 2022, we met for the first time again in person in Brussels in November last year. So I remember the welcome note from the kickoff meeting that Frederic and I have wrote in 2017. And there we have referred to avian H5 and 8 influenza viruses as one of the major episodic threats. And then we have seen the rampant spread of African swine fever and periodic outbreaks of zoonotic flavivirus infections. Then, of course, there was COVID. And today, in 2023, avian H5 influenza viruses present already a bigger threat than ever. And during all this time, we have been working at the front line of the European research response to these episodic and zoonotic threats. So this shows how important our activities actually are and the importance of our network. And the objective of our meeting today will be to look back at our achievements during the past 6 years and also to discuss our future. So we have a complete workshop dedicated to sustainability tomorrow, where we will discuss all this. But before we will talk about our future, it's very important that we center our attention on the now. And I will ask now Anthony to brief us about the actions that are expected from all of you during the next weeks to come. Okay.
Okay. So this is a very short speech about management, and then you're clear with that. So this is the project timeline. The project was extended for 1 year, and we are coming slowly to the end of the last month of the project, and which means that we will have to do a final reporting of the project. I sent you all yesterday the information to complete this reporting, and we will use a lot the SharePoint platform of the project to collect all the information. So it's very important that if you do not have an access to the SharePoint, you contact me, because the platform link changed between last reporting and this one, so you might have to reset your account. So please contact me if you have some trouble to reach the platform. We also still have some work with some deliverables to finalize. We have 27 deliverables that are in preparation. And we also have some transnational access short reports that we have to complete for each either installation or partner. Some partners have several reports to produce. All the templates have been included on the reporting page of the project on the SharePoint. And again, if you have any question regarding that, you can ask. But those TNR reports are very short and only contains information that you probably already provide. So it should not be very time-consuming to prepare. Then for the next action regarding management, we will review all the last deliverable to be submitted by February, including the TNR reports. We will compile all the technical reports, financial reports, and the dissemination reports within March 2023, because the submission of the final report need to be done 60 days after the end of the project, which is April 2023. Then we will probably have a project review meeting with the war package leaders, probably in May. I hope that we will be informed as early as possible, because as you know, the costs are not eligible outside of the period of the project. So if the war package leader want to be reimbursed of their cost, we need to plan the travel cost as early as possible. And then we might still be in contact if, for example, some deliverable needs some revision. Then we will contact you again during the year in order to finalize the project. And this is for the management. Now I will leave the floor to Sasha.
Mabel, she will present the agenda. I just want to specify for the ethics deliverables, what we request from you is essentially all the authorization numbers you have for your facilities and the ethical licenses. This is what we will request from you very, very soonish, that you can already prepare that if possible. And it will be compiled in 3 different deliverable reports. They are a bit redundant, and we try to compile it in a table where everything is listed.
So regarding the agenda, so today we are glad to have the presentation of all the TNA projects by users. But before this, we will have a presentation from Norbert regarding the TNA access management, a general overview of the TNA. Then we are glad to have Jens Langermann, who took part of the USP selection, the user selection panel, who will provide his feedback. And then Arnaud Bataille, Gaetano Donfrio, who is here, Jean-Claude Sicard, Dolores Gavier-Waidon will present that work. There will be a break for the lunch around 12. And then 3 other presentations of TNA project, Caroline Denèvre. Okay. Sandrine Le Cellier, Lucia Vasquez Salgado will present that work. And we will finish by a conclusion by Norbert. After the coffee break, we will have 2 other independent presentations. The first one by you, Simon, regarding the best practice part of the project. And then Kate Miller will present us ethical aspect, free air, and social impact work that has been done within VetBioNet. So I think now it's Norbert.
Yeah, it's Norbert.
So nice to be back in tour after quite some years, 6 years, and you already mentioned it already. And I saw we did not, time was going further, but apparently we escaped changes. So welcome everybody. I would like to talk about the transnational access, and I did not follow the recommendations by Anthony how the presentation should be, because I think for the last meeting it should be good to have a more overall look at what we have done within the TNAs and the transnational access. I call it a central part in the VetBioNet infrastructure project, and I'm pretty sure that we all enjoyed in a certain way who participated in it from these TNA activities and the opportunities we got within the funding for this type of activities. I would like to thank also Astrid and Rineke and Marlise, who joined me at WBVR to organize it as a transnational access point. And that was always a great cooperation. But let me thank on forehand already on all others, on your participation, on your support we got in preparing the transnational accesses. And in the following, I will also mention the selection panel who was very much involved in it. And I haven't seen Juan in here. He cannot make it. It's a pity. And I can tell you, Astrid and Rineke yesterday in the train decided to go back because France is not a very reliable partner in traveling. So, but here is a picture. You will see it tomorrow. Sasha will use the same, but I looked at it and it's very nice. It shows all the partners who are participating in the transnational access. And for those who are not familiar with this, we had 15 partner organizations for this infrastructure project and 26 installations, which give us a very broad field of possibilities to provide access services. What did we offer? Just to remind you, we offered animal biosafety level 2 and 3 commodities and laboratory facilities, and also the validated, more or less validated, I have to say, animal models, because we also were quite flexible in the different projects we were using. And we also developed animal models, or we used animal models for DNA projects. We also had the strengths to offer disease monitoring tools, imaging facilities, and access to tissue and serum banks, pathogen banks, and study design expertise. That is from one of the first slides we presented when the start of the project was, just to show what we could offer in the project. And most of the issues were also used within the project period. The logistical achievements we had in this whole period was that we started very early with a web-based interface to launch the transnational access call, and that was done in cooperation with EAAP. And we had some trouble in the beginning, but in between we improved the accessibility, so we were able to have a good online and a digital communication platform to provide the users and also the service providers and also the administrative part with all the information from the transnational accesses. A very important part was the independent user selection panel and the TNA evaluation process, which was established very early. We could build on what we have had and experiences from the Nadir project, which was quite good. We also had luckily some experiences with other infrastructures so that we could learn from each other how infrastructures could be organized and what we could, how we could improve the process. And we decided also in the project to have an easily accessible front office structure, the transnational access point, which was located at our institute. And with this, I think we had a very clear structure to find the transnational access and to provide the information across the partners. The user selection panel, I want to mention specifically, and Jan is so kind to give us his retrospective on it later. But in this, we had 7 highly experienced scientists from outside the consortium. That was the important point, to have an independent view and review of the projects from different expertise, and the names are listed here. And additionally, we decided to have an internal USB part which supported the review process by giving information on animal studies, on feasibility of animal studies, for example, and also on ethical points. And we had always very nice discussions, in my opinion, on the different projects, and we tried to improve the projects, try to pinpoint the difficulties when there were difficulties. And I have to say, the common efforts were really great, and it really should, some tulips from, I would have brought some tulips from the Netherlands, but it's too early in the year. Then we decided very early the eligibility criteria, and I do not want to go through it, but what you see is that we in first instance had the BSL-3 pathogens as a major issue in zoonotic pathogens, but also we decided also to work with BSL-2 pathogens, which need to be worked in high containment facilities. So we realized already in Nadir, but also later, that the high containment facilities are not only necessary for BSL-3, but also for BSL-2 pathogens due to their pathogenic characteristics, but mostly due to the fact that the studies need to be done in contained environment to not compromise the study in itself. And we do not have so many in Europe. So this is quite important to mention also. Then we have the TNA access point. Yeah, that was the idea. And I think we were quite, I think we were quite successful. You never say it about yourself, but there's a one-stop shop we were able to create. We had an overview of the services which could be provided. And so we had a sort of a front office function where we were coordinating the user selection panel. We were coordinating the work to the partners. We knew about the animal models which were available, discussing with partners if there was a request for a certain study, which partner could do this work in time. We discussed the study design and also advice on the study design to the user projects, which were the customers. So it works quite nice. I think this structure is for an infrastructure project a very advisable to have. I think in Isidore, we have a comparable system, but yeah, we should discuss these experiences and experiences of other infrastructures also in the Isidore project to recommend maybe how to what is advisable to work, how it's advisable to work. Just showing the transnational access process. Most of you know about it. It's the submission of an application abstract. The TNAAP access team controls the eligibility, searched for the infrastructure, and that was usually around 1 to 2 to 3 weeks. Then the full submission was requested. That was prepared and in the following USP discussed. Intended, there's also a very old slide to have it in 4 weeks. And for a number of projects, we were able to have it in 4 weeks. For others, it took longer, but that was also part because you're also dealing with the user projects. We have to react on it. Yes. Yeah, I'm working in different infrastructures, and it is quite unique for VetBioNet. And personally, I think it's very recommendable to have a very early contact between the user project and the service provider. And the reason for this is that it increases the feasibility of a study. It increases the insight in what the study will cost and how the schedule of the study could be, because we are not working in a time-free zone. We are working in a project period. So if you have a project period of 4 years, you need to be quite quick. We are talking about studies which can take quite a long time. So this remark by Sasha is very important, that an early interference between the service provider and the user project is very important and shortens the process. Then on the right side, you see we have the evaluation period. This is the user selection period, the user selection panel, which reviews the project and gives an advice to the executive committee, so the responsible committee of VetBioNet, whether we should proceed, whether VetBioNet should proceed with the study or not. So this was a quite rigorous procedure, and by this flow, we could minimize the time for the project evaluation period. I have to say that we have projects which were within shorter than the expected 8 weeks, and we have projects which took quite longer than the expected period. But I think this is evident also due to the complexity of projects. And between January 2018 and 2022, we had a total of 77 TNA requests, and we had 51 proposals approved. And one approved project is just withdrawn last year due to the fact that the feasibility of the source of the requested animals was not granted, and there were some struggling with the project in itself. We had withdrawn 26 proposals, usually due to lack of eligibility, feasibility, scientific soundness of the user himself withdraw this. But there were also always quite some reasons, and they were also always discussed with the user selection panel, so that we had a continuous up and down of discussion between the user selection panel and the transnational access point. What we did, as I said, we looked at the pre-proposals at the access point. We discussed the proposals and managed a total of 25 online user selection panel meetings. We would have liked to have at least a couple of them on face-to-face, but the COVID period did not allow this. And then we learned how efficient online meetings can be. And therefore, most of the meetings were online. We tried to keep track on the progress of the TNA projects. We requested the information by the service providers. For example, of the ethical license, we were keen to have the information on all the ethical improvements which have been done and which is necessary also in reporting later to the Commission. And we chased applicants about the project to give information, project status and reports. And that would, especially in the last period, it's very important to chase and to get everything in time for the reporting. Users of the projects, this shows where the users come from. We have users from 21 different countries, which is, I think, really amazing. And most are from academia and research institutes. But 4 users were from small, medium enterprises, which is quite nice. I really like that we were able to have this mix between academic and research institutes and the private partners. And the principal leader was always from a user project was also always outside from VetBioNet. That is a requirement of the infrastructure projects. But I also have to say that it's sometimes or relative, that's quite common, that there are already research bonds between the partner and the user project. So this, the network, and our network is quite small, that has some important influence on the use of the infrastructure. But sometimes these are starting points of new corporations. I think this is also something I really would like to stress, that it broadens your network to be part of an infrastructure. If we look for the gender distribution of the user projects, it was 40% female and 60% male. But this is especially for the first name. Later, when we have all the reports, we get the names of each participant, and that will probably change the percentage of male and female, which is shown here. Another nice thing and very colorful is what we can offer to the research community to work in very different species. And you see here from just a cell line to cattle, swine, goat, sheep. In fact, all the species this type of institutes are working, and they were requested from the VetBioNet. But I think this is the strength that we could offer this type of work in the broadness of the zoological landscape. And if we are going further to the pathogen classification landscape, we dealt mostly with pathogens classified according to the EU Animal Health Law, and especially to the A, D, and E category, which is the highest categories. That was the 41%. 8% was category C, C, D, and E, and D and E, which is the lowest virulence characterization, was also in a few, and 41% are not categorized. That means that these are not in the list of the animal health law, but nevertheless important, either economically important or from infectious standpoint important pathogens. Virus research, which was requested in regard to influenza, African swine fever, foot and mouth disease, and you see the whole list, a very different list of pathogens which were dealt with. So this again shows the broadness of this network in terms of availability of animal models and expertise to work with certain pathogens. This is a list of bacteria we worked with in different projects and parasites. So again, this corroborates the broadness of the whole infrastructure. And if we type the studies in objectives, most 38% were viral pathogenesis, 20% were immunology-related topics, vaccine development 18%, and bacterial pathogenesis 14%, 8% parasitology, and 2% antimicrobial resistance. So also the objectives of the studies were quite different and also representing what our institutes are covering in the broadness, but also which is requested in the field. Now we're coming more into detail. These are the reported studies. I do not want to go in detail through the studies, but just want to show since the type of studies, the title of the studies, these are reported. And we will see a number of these soon in the TNA presentations later this day. And so we have 29, I think we have 29 reported studies, and we have 5 studies which are completed, which I expect to report within the next couple of days. And then we have studies which are in execution, and this is a little bit of concern, of course, because we are already on the 7th of February, and we still have a number of studies which are in execution, and this is something we have to discuss here, that these are really finalized before the 28th of February. We were confirmed that the numbers are finalized, but we have a few which are still questions. I already discussed with Sandrine about it, and she confirmed that it will be finished by the end of the month. Ignacio Garcia, this study is done at APHA, and that is a little bit, I'm not sure that where we are exactly, because the correspondence with the user project is not always easy, but we will discuss it later. And there's a HMSIT, which is a very nice study insofar as this really showed the cooperation between different users, different partners in the VetBioNet infrastructure. Usually we have studies which are done, requested for one partner is doing it. It's not necessary to be, in fact, in the VetBioNet consortium, but this one is a collaborative effort of INRA and of Roslin, and that has to, and I think the samples are in Roslin at the moment, and I need the confirmation by Roslin that it will be done in time for the reporting. And this is just an example of what we did in between. We sent a questionnaire to all the project users, user projects, to give their idea and their opinion on what they have with the VetBioNet project. And in regard to the projects, this shows that the enthusiasm of the users were quite high. There, yes, the data were in line. You see the graph underneath that shows that a very high percentage was agreed with the results, but this was in a very early phase of the project. And but what you see is that most data was as predicted, but there were exciting new aspects. That is a very nice comment of one of the user projects, or we could perform a study in lambs, which we could have done without the VetBioNet project. So the gratefulness of most of the users is very high and very nice to read. In summary, we have 58% reported studies, 10% are finalized, and 32% are in execution. So in the remaining months, a lot of work has to be done. 2/3, most likely no problem. Studies and executions need to hurry. And in the feasibility of 2 studies we just discussed. So this month is a month with a lot of work. So I do not know how could we have a 3-day meeting when we have to do such a lot of work. This shows the allocation of the TNA services to the different service providers, and winner is INRA IRB with the most of studies. And I'm really, unfortunately, Bertrand couldn't make it, but they did the most of studies in fish and on the mammalian species and avian species. APHA, Hugh, many thanks to your institute. You're the winner of this. And IRTA did their best, but within the budget. So a lot of, you see a quite nice distribution of the services across the different institutes. And many thanks to all the colleagues and the key persons at the institute we had to deal with in the startup of a project to discuss and to get information. That was really nice cooperation. We really enjoyed it. And also the service providers were rated very high. You see here is a rating and a rating of 1.6 for the timelines, the satisfaction 1.1. And although the COVID pandemic, the service provider had to do more hands-on work since my team wasn't able to travel to help. This is one of the comments. So the overall impression of the user projects about the service providers was very high. So very many thanks to all of you who have contributed to this success. Financial aspects, I will keep it short because this is, I showed it already in November to the community, and I do not have additional information. I think we have more or less used our budget to the point, but we will see it, I think, in end of March, whether this is true or not. So, and the financial part is more or less in hand of Sasha. So I'm happy that I do not have to do this. So milestones, and this comes back to the template we get from Anthony. Most of the milestones are, and my deliverables are done. 1.5, no, this one, the 1.7 was open. That is more or less closed today. And we have the results of the TNA request evaluation. That is the last report, this pending for the last report. So I think in terms of deliverables, we will make it from the work package 1. In terms of joy we had during doing this work package was quite high, or the pleasure factor was good for us. So thank you everybody for participating in it, and I'm really looking forward to the presentations of the TNA user projects this morning and this afternoon, because I think they can give a very nice impression to us from the community of VetBioNet, but also I hope to all stakeholders who are also online or in the room to see what we have achieved with an infrastructure project, which is a quite important one. So many thanks to all of you. The VetBioNet Transnational Access project is now closed forever. So we showcase now, I'm not sure where we are in time, because... Oh, okay. No, first of all, we have... Yeah. Okay, are there any questions to the TNA, or to how to proceed further until the 28th of February?
all questions regarding reporting, administrative aspects, yeah, just contact me and Anthony that we can settle this as soon as possible.
Okay, but then I would like to hand over to Jan. And Jan is the head of a user selection panel, has chaired this for the last 6 years, and was always in a very, for my part, in a very nice communication, very efficient communication. And so I'm really looking forward to your review on the time.
Okay, thank you, Norbert, and thank you for the overview. I don't have any slides, so let's just give you a feedback on how the user selection panel acted during the last 6 years, which was a year longer than anticipated. But I must say, first of all, that we could not have done anything of this as efficient as we did without the great help of the TNA access point. I think that was a really important part, that the TNA access point always had everything in place, and we had very good and open communication with them all the time. And that's really essential if you want to work efficient as a user selection panel, because we did have 25 meetings, something like that. So there was quite a number of meetings during that time. Well, one of the good things of this VetBioNet, I think, with the TNA, with the access, so the TNA access, is that there was a really good combination between the external user selection panel and an internal panel, where we had a continuous communication, and we were both there all the time. And I think that after 25 meetings, we have had perhaps once or twice that we didn't find quorum, so we had to recancel. So all the time there were enough people there, which already shows that there was enough interest from both parties and enough enthusiasm to judge all these proposals that we received. And of course, one of the good things is always that there is first an eligibility check by the TNA access point, so we could work very efficient. And I come back to one of the things Norbert stressed, and that what is very important, and we noticed that as well during the judgment, it is essential that we notice that there was already contact between the access requester and the provider beforehand, because you can notice that if we knew where they were going to, what they wanted, there was a question, do you know where you want to go to? And that's essential if you want to judge it in a good way, because if you don't know the access provider, then it's very difficult to judge whether they can offer the right access, or you can have the combination with the requester and the provider in a good way. I'm myself also sometimes on the other side with other projects, and if we notice that some people come in with requests that you think, well, we cannot do this. This is ridiculous. We cannot do this. So it's essential that you know beforehand what requests are. So we had a very interesting group. It was also very challenging sometimes. Norbert already showed there was a lot of different species that there were requests for. There were a lot of different pathogens that there were requests for. And we were a small group of 7 people, and then the internal user panel, which needed to judge whether this was all feasible, scientifically sound and feasible to perform. And there were quite interesting discussions sometimes, I must say. The way we organized it and was organized was that we had 2 people of the user selection panel, of the external, did have a more in-depth look at the requests, and they prepared a report, sent it back to the TNA access point and to all the panel members, and they all read it. And then during the meeting, everyone had read all the proposals, and then they were discussed. But the first were the 2 people that started the discussion, and then there was a general discussion. And both from the external members, there were sometimes very good feedbacks, or very often good feedbacks, but also the internal members had very good feedback sometimes. So that was a very nice and open discussions always. Sometimes we send them back, or at least very often we did send them back with some questions. Sometimes they were immediately approved, and sometimes we had to reject because of not feasible or scientifically. There were so many questions that we said, now, well, this does not fit within this project, so that we need to redo that. I think that's most of it is that it was really fun to work with those people. It was it took a couple of hours at least. Each meeting took at least, I think, 2 or 3 hours, 2 hours at the minimum. So it was something. It does ask some of your time. That's another challenge, I think, for future projects. You ask people to do this voluntarily. You're not within the project. You're not paid for it. You do this for free. You need to like it, and you need to have an expertise in judging those projects. So that was, but we had a very good group to do this. And I must say there were very few people that skipped some of the meetings. There was not, sometimes of course you cannot make it, but that makes sense. What was also very important is that with the TNA access point, at the end of each project evaluation, there was a summing up. And we did that for each project immediately. And I think there was the expertise, I would say, from both Astrid and Rieneke that immediately made a summary. We repeated that at the end of the proposal discussion. So there was already ready by the end of the meeting. They could send it back to the, almost immediately send it back. And it was checked immediately during the meeting by the user selection panel. That worked perfect. And that was really, I think, amazing how fast and how efficient that went. I'm also experienced with other user selection panels, which I must say took much longer sometimes to get some feedback. So I think that's a big thanks to all my colleagues from the user selection panel, the external ones, that unfortunately could not make it. I know I saw Patrie online, I think, but it's Juan should be here, but he couldn't make it, and some others also not. Also, a big thank you to the internal USP members, because it's also very valuable feedback. And also many thanks to the program that we were able to do this, because I think we really liked it. And DNA Access Point, thank you very much. So unfortunately, it comes to an end. If there's any other projects doing this, it's great, but I would say also Easydoor or whoever, go to VetBioNet and ask how to perform TNA access discussions, because I think this was a great example on how to do it efficient and also workable for external members. So thank you very much.
The access point is closed.
I would just like to thank again Jan and the whole USB and the TNA access point for the fantastic job. Especially, we have to keep in mind, as you have said, you were not project partners, and 25 meetings, 2 hours of time. That's a huge commitment, just in-kind contribution. That's fantastic. A really great thank you. I really think we should applaud there. And then another thing I would like to highlight this, well, the work of the user selection panel was not only to evaluate projects, it was actually in the end to improve projects. And if you look at the figures that Norbert has presented, I mean, we had 77 TNA requests. I don't know if that is pre-applications or full applications, but anyway, in the end, we had 50 projects granted, and that's in a very high approval rate. that doesn't mean that we just waved everything through, but on the contrary, that we really tried to improve the project. And of course, it helped that in most cases, users and access providers were already in contact. And then all parties could then contribute to the improvement on the project. And I think at the end, we just had high quality projects. And once again, that's thanks to you all. And I would just, you mentioned it, I have to comment on this. The wrap up from Astrid and Rieneke, that was just amazing. That was really immediately after each meeting, after each presentation of a project, it was printable. So I'm sorry that you cannot be here, Astrid and Rieneke, a huge thank you.
I'm Martje Venten of Vlissingen. My badge was printed after all. I think it's time to learn from this experience. The lessons learned are to be very important in future EU projects. And having said all the good and proper things about the user selection panel's functioning, it would be really important to understand how people become aware of transnational access programs, how they can ready themselves to submit a proposal, who to contact, what's possible, and looking at the type of mechanisms behind it. I think many people just don't believe it. It is too good to be true. So there are few really novice users. We have here all the promotion materials of EasyDoor. EasyDoor, again, is mostly based on transnational access as a way to push collaborative research, but also the management of contagious disease outbreaks. And are there any lessons that we can teach, lessons learned that we can teach EasyDoor's functions?
Yeah, well, you're correct. There's more now with EasyDoor, for instance. There will be more TNA programs, I think. One of the important lessons is still, I think it was mentioned a couple of times now already, is it's so essential that the ones that request an access also first have contact with the access provider, because if you don't have that, it's you can end up with things that are not realistic, or that in the end look very nice, but that's just not doable, of that the access provider has to say, no, we cannot do that, or whatever. You need to be realistic there. And it's essential that beforehand there's at least some contact. Now, there's of course always a challenge how to do that, but it's because that's another issue. So there, a TNA access point might be in between there or whatever. But that for me was an important lesson, at least, that if there is no contact, then it's very often doesn't work well. And what is always a challenge as well is to get sufficient people that ask for access. Now, VetBioNet, I think, has been exemplary, that they have enormous number of projects, but sometimes you also struggle. I know there's some of those projects that struggle with getting enough people there to have to ask for access. So advertisement or mouth to mouth or whatever, you need to be very open, and that's, you're correct. Some people find it too good to be true, and that's how to make it clear. Kate has a remark there, I think. Yeah, you need otherwise at home they cannot follow us.
sorry, just to quickly add, Marta, really important points you raised. And just to highlight that in my session, right at the end of the day, I've highlighted some important dimensions of the VetBioNet work that relate to our ethical and social dimensions. And of course, our raison d'etre is TNA access. And I think at this point, Norbert has identified a number of sort of key reflections, and I'm going to set out some boards to capture your sort of final reflections as providers and members of the network. So please, Marta, add those comments to the board, because I know that Norbert is going to take those comments that we can capture later and use them in your final report. So, you know, please, these reflections here, don't lose them this morning, make notes and write them down at about 4 o'clock this afternoon. And online members will provide you with a space so you can add comments as well.
I think, Matti, you raised 2 questions. The one is how to address and how to make it believable that there is such a possibility. And in my experience, I presented at 2 virology, European virology meetings, the project, either in a presentation or in a booth. And sometimes it was really like you saw the unbelieving in the eyes of the people, and that is which maybe overcomable at the moment if you show that you have done this type of studies. So the showcases we have this morning are very important for the community to show what possibilities are within this infrastructure project. That is an important part. What you also learned is that the internet advertisement is of a relatively low success. It's mostly mouse to mouse, at least what we experience, and the spreading of information within your network. So this is a little bit double, because that makes that looks that the service is available for those few who know about it. And this is something we principally do not want to have as public money. So what we are saying is the advantages of having a very close interaction between the service provider and the user project and the allocation of a project to a user project is a very delicate one. If you want to be in time on the one hand side, if you want to be have it done in the best way, and if you want to be sure that there is no conflict of interest. So it's always a quite delicate question we are, and therefore it's very important to have a very responsible user selection panel taking part from outside and to have a look at it. This is a key factor in being clear that there is no conflict of interest. But we are a small world. This is also something. We are in a veterinary world, mostly in the biomedical world a little bit, but mostly in the veterinary field, and that is on forehand a very limited field. So we have to be very careful in how to respond to the public interest that we spend public money in a decent way.
I just want to say a few words or comment on the EasyDoor issue that was brought up. Well, of course, it would have been the ideal situation that each infrastructure that is participating in EasyDoor could have kept its own selection mechanism and workflow. And however, the problem was that we had to come up with a harmonized application process with a web interface that precludes at this point normally that users and access providers are already brought together. However, this is not something that we exclude. On the other hand, it is possible. Jan, you know it. you are access provider in one case, and you have already been brought together with the access provider. It is possible, and that's something that we can discuss also with our Erinia colleagues and other participants in EasyDoor. I would also like to welcome David, who is here as one of the representatives of the EasyDoor infrastructures. He's representing EATRIS, and we discuss those things tomorrow then.
Any other comments or remarks? If that's not the case, we are a little bit ahead on schedule, and no, it's oh yeah, I know it's yeah. We did not make any agreements on who will moderate this next session. I will do it. In any case, I will take the first one. And I think Arnaud is already online. And yeah, if you could bring the Arnaud present. Arnaud is online also, I think. Yeah, okay, thank you very much. Okay, and he will present online. No, Arnaud will present online. Yeah? Yeah, okay. Just you see the program, and I have to apologize to Lucia that and also to Sandrine that I made a mistake in my slide on the time points, but you will have the exact time points in the exact agenda. What we looked for is to showcase which type of accesses we had and the access requests we had. We made a selection of between different species, a selection of different pathogens. So you will, when I was showing the tulip, the flower of the tulip, you will find more or less in these showcases. But what you also will find here that the participants within the VetBioNet infrastructure were able to provide this type of accesses. And I hope that the presenters will also give a little bit an insight how they experience the collaborations. And we have some which are presented by the access providers. Sandrine will provide a presentation. And it shows also the close interaction between the user projects and the access providers, which is necessary for a successful project. So I would like to invite as a first speaker Arnaud Bataille, and he will talk about his studies of virulence of Peste petit ruminant virus at the Swiss Institute of IVI. I'm not sure whether there is a representative of IVI present in the meeting or online. No, okay, but Arnaud, the floor is yours.
Thank you. Good morning. I will need to have access to share my screen. I do not have the right for the moment.
Can you do it? Do you need?
No, I still can't share my screen.
Okay.
And now it's good. Can you see my screen and hear me well?
Yes.
Okay, great. Good morning, everybody. Thank you for giving me the chance to present our work on the study of virulence of Peste des petits ruminants virus in relation to the variability of host response. So it was work, as was said, done with the Institute of Virology and Immunology in Bern. Just first, maybe just a quick background on the history of this collaboration. So in our case, we had already first exchanges with Arthur Sommerfeld of the IVI in Bern for possible collaboration, and it was actually before the knowledge of the existence of this VetBioNet call and their involvement as facility provider. And we received first information about the VetBioNet call in January 2018. This is coming from our research unit direction at CIRAD, and from there contacted back Arthur to start preparing a proposal. So we did submit a pre-proposal, and then with them in 2018, after a couple of revision requested, the project was accepted in August 2018. Along with this funding, I had a PhD grant funded through our program as EU reference laboratory for pest deputy ruminant to work on that project, and the student starting in September 2018. Just as I mentioned, we are still collaborating with IVI in analyzing really the last data of the experiment, as I will show you. A quick background on the virus. So pest deputy ruminant virus, PPR, for its abbreviation, is a morbillivirus infecting mainly sheep and goats. It's widely distributed in Africa, Middle East, and Asia, with some incursions in Europe, notably in Bulgaria in 2018. And there's a global campaign of eradication of this disease organized by FAO and the World Organization of Animal Health, with a target of eradication by 2030. So within this kind of frame, one main question for the control of this disease is about the host's susceptibility to infection. It's been shown in the field, notably, that the susceptibility to PPR is highly variable. It depends on the species infected, but even in the breed, the hosts, and also on the virulence of the strain that is circulating. So different strains will infect differently same hosts. And we don't really understand the mechanism behind this variability. So before starting the VetBioNet project, we actually already had quite a reliable experimental model to study this from a former infection experiment, where as a model we had the Saanen goats and different strains of virulence that was quite variable in that specific host. We had one that was a low virulent strain coming from Ivory Coast, called Ivory Coast 1989, and another one from Morocco called Morocco 2008 that was highly virulent in that host. And then we have a very well-known and widely used vaccine that's a natriurenated vaccine called Nigeria 75-1 that provokes no symptom at all. It's totally innocuous. So here we use this model to test a hypothesis that the modulation of the host immune response during infection in small ruminant can be explained by variation, can explain the variation in pathogen virulence and host susceptibility. So we went in designing the project in 3 phases. That's how we presented it for the VetBioNet project. Phase 1 as an in vitro experiment that started in May 2019 with an objective of first exploration of the differential gene and protein expression in immune cells infected by the different PPRV strains. So there we would use PBMCs collected from blood and then infected either by one of the 3 strains, and from there do flow cytometry, RNA sequencing, and proteomics. In the phase 2, we would then move to an in vivo experiment. This one started in August 2020. There, the objective was assessing if we had similar trends in a more natural settings in terms of gene and protein expression for the immune cells that were infected by the different strains. So there we had different groups with 5 goats infected by the different strains, and with a control, and they're collecting blood to collect the PBMCs, and then do again the flow cytometry, RNA sequencing, and proteomics. And in the last phase, so another in vivo experiment that one already started in May 2022, so it was recently done. Here, the objective was really to look inside the target organs, because the PPR virus is kind of picked up during in the respiratory tract by the antigen presenting cells, and going there first in the tonsils, and then in the from there being infecting the immune cells that will then spread it towards other lymph nodes and other organs. And we wanted really to look at early stage of infection in the different organs. In that case, we had 3 groups with 6 goats infected by either the low virulent or the high virulent strain, and the control. For 3 goats, we stopped the infection experiment at day 3 post-infection, and for the 3 goats within each group, we stopped at day 6, and from there collected the organs and the blood to do the different tests. Different organs were going to be used for different type of transcriptomic analysis, like the tonsils and lymph nodes for the bulk RNA sequencing, and we have some organs that will be used for single cell RNA sequencing, others for flow cytometry and pathology. So we'll just quickly present some results, first from the phase 1 for the in vitro experiment. First, looking at the patterns of viral replication of the different strains in the PBMC, and this is done by detecting the protein. And with the 3 methods, so with flow cytometry, with RNA sequencing, and mass spectrometry, we really have each time the same pattern with much higher replication of the high virulent strain, the Morocco 1, compared to the 2 other strains. That was quite as expected. Looking at which the number of genes that were differentially expressed in the different groups in terms of upregulated and downregulated, we had many more genes that were detected as upregulated. Some were common to all groups, but many were specific to the group with the high virulent strain, and others were shared or unique to the other groups. In terms of downregulated, we have mainly only for the high virulent strain, where we did see a lot of downregulated gene, but not for the other groups. And looking more quick in details of what were these genes and which type of immune pathways they represented. Just quickly here. So every time you have the dot in red, it means that it's upregulation, and in blue, it's more like downregulation. The main results were that we had common to all strains an upregulation of the antiviral response, and with response for the vaccine strain and the low virulent strain, Ivory Coast 89, broadly similar, including capacity to inhibit the viral replication. But we had many more pathways that were specific for the Morocco 2008 strains that were either down or upregulated, notably a heavy inflammatory response. So looking at proteomic analysis, there we didn't see so many differences between the 3 groups, mainly saw proteins that were associated with antiviral response and that were differentially expressed for each group. So this phase gave us first vision of the mechanisms that are involved in the infection of activated PBMCs with things that are like common without for all strains. So with some activation of the antiviral response and inhibition of proliferation. But when you have a high virulence strains, all this is much more, the response is much stronger inhibition of proliferation, of stronger apoptosis, and also a whole cascade of inflammatory response. So we wanted from there to see if we had the same patterns in an in vivo experiment. First, we really took the chance to really look in details in this comparative pathogenesis of different strains in salmon goat, because it was had not been done before. So we could confirm that the different viral strains showed the same virulence patterns in this model as was shown before. So it was as expected. Interestingly, we had a significant decrease of lymphocyte counts, mostly for the Morocco 2008, associated with really the inhibition of lymphocyte proliferation and killing of those cells by the virus. But some things were unexpected and quite interesting for us as diagnostic lab for this virus, is that we could detect genetic material even of the vaccine in all organs and ocular swabs during infection. And this is really the first time such a detection is possible. So it was quite important for us to be able to do that. Also to be able to detect genetic material in serum for the Morocco 2008 and Ivory Coast 89, so for the two strains. Then really looking into, again, the gene expression, so really, again, multi-omic approach to analyze the immune cells response to infection. So we had collected blood samples to collect PBMCs at different time points, for the different approach: flow cytometry, RNA sequencing, and proteomic analysis. Sorry, this is not very sharp, but again, looking at gene regulation, had interesting patterns in terms of genes associated with innate immunity. So at day 7 for the Morocco strain, we had an enrichment of the antigenic presentation. For all strains, we had similar interferon-1 response due to the presence of the viral genes. So that's more like what we see here, the similar response of interferon. And just for Morocco 2008, we had like all the upregulation of the inflammatory response as was seen in the in vitro results. So really showing in this natural experiment, well, natural setting, same results than with the in vitro experiment. Noting for the adaptive immunity, we had similar trends for all strains. So it's really with the innate immune response that we could see differences between the different PPR strains. Here we had also a more complex results in terms of proteomic analysis. In all cases, we saw proteins that were related to the interferon-1 response, as was seen in the proteomics. But for the Morocco, also we had a whole set of proteins and interactions that were linked with inflammation and ribosomal proteins. So that was unique to the highly virulent strain. So with the results of these first two phases, started to have some conclusions really clearly capacity of the different strain to replicate in the lymphocytes really differ quite sharply. And it is probably why we have different levels of immune response and notably the inflammatory response appearing during the infection with the high virulence strain. And this capacity of high viral replication and high interferon-1 response seems to be key factors in the capacity to inhibit lymphocyte proliferation of PPR. And we have a clear, stronger innate immune response activated when you have a highly virulent strain, while the adaptive immune response appear to be more or less similar between strains. So just quickly presenting the first results of the phase 3, as I was saying, it happened in May 2022. So we have results from the RNA-seq, but all the rest have not been done yet. But really, we have all the samples, all the data, and now we have to kind of do the last sequencing analysis. So first question was detection of the virus genetic material in the different organs at day 3 post-infection or day 6 post-infection. And as expected, because tonsils are like the first organ, the first target of the virus infection, that's where we'll find stronger presence of the virus and increasing with the days compared to other mesenteric lymph nodes or in the white blood cells. But in all cases, each time it's increasing from day 3 to day 6 for the highly virulent strain, while for the low virulent strain, we actually started to pick up the virus from day 6 only. They're looking at gene regulation in the different organs, and first looking at genes that were associated with innate immune response. It was very interesting to see that the patterns of upregulation of the antiviral response and inflammatory response really seems to differ between across organs with for example, here for the tonsils, you have the inflammatory response, but you actually don't see those upregulation for the lymph nodes. And in terms of the response for the Ivory Coast, the low virulent, but as expected, we started to have upregulation a bit delayed. So from day 6, having the antiviral response, but also with no inflammatory response. So as was seen before, but with patterns slightly different between organs. And also very interesting is something that we will explore further is differences in terms of gene regulation for the adaptive immune response. So it was clearly more like downregulation patterns associated with the inhibition of cell proliferation, and those patterns really differ between organs. So maybe different capacity to inhibit cell proliferation in different organs, and also for different strains. So it's something we will look more into. So as I was saying, still to come, well, we have to finish the analysis of this RNA-seq for this experiment. We will get started with our colleagues at IVI on the single cell sequencing of some samples, doing multicolor flow cytometry and digital histopathology to compare results between the 3 approaches in terms of picking up the different immune cells populations and the presence of the virus in those different populations. So really, in terms of general output for us from this VetBioNet project, it's really first was very important in terms of capacity building, in terms of immunology and virology research here at CIRAD. Those type of research in terms of immune response, transcriptomics, and sites was really new for the PPR team. So we really started, and we will keep on new lines of research thanks to this to the VetBioNet project. We definitely build up our capacities in terms of flow cytometry, transcriptomic, proteomics, and also in terms of design of projects involving animal facilities, and allowed us to involve many PhD and master students in the project. Very importantly, it really built up strong collaborations, not only with the IVI in Bern, but with other research institutes in those questions that we will keep on working on. And definitely also some outputs that were maybe not what we, the direct objectives of the project, but definitely some outputs that lead to maybe new methodology, new diagnostic tools, notably capacity to detect PPR in different tissues, from now exploration of genetic markers of variability of in immune response to PPR, and looking at, for the moment, at developing in vitro immune cells infection assays to assess the host susceptibility to PPR strains. So trying to, from there on, limit the need for animal experiments when they are not needed. So just to wrap up, of course, there was an important team that was involved in this project at CIRAD, also at IVI, also collaboration with the proteomic platform here based in Montpellier for the proteomic research. And of course, a big thank you again to the VetBioNet project for funding this research. Thank you very much. So I cannot hear you. Huh?
You cannot hear us?
Yes, now it's good.
Now it's good, okay. Many thanks, Arno, for this very nice presentation. I think it was one of the first projects we granted, and it's a quite long period. It's a very diversity of methods which are used, different approaches in the study design. So it's a very comprehensive study. So first of all, questions from the audience to Arno. Before the lady.
Thank you very much. This is Julia Vergara-Ler from IRTA CRESA. And I had a question related to, well, mainly phase 1 and 2 experiments that you mentioned, but it's also linked to your last conclusion. So first, in the in vitro experiments, did you use, maybe you mentioned and I didn't hear it, but did you use PBMCs from different animals? And also, are they from the same breed of the animals used in the in vivo experiment?
Yes, yes. So the PBMCs are all coming from the same model, the Sand goats, and each time for the replicates, it was replicates coming from different animals. So.
And I guess you didn't see differences between hosts, either in vitro or in vivo, or at least significant difference?
Difference in the patterns of in our results between in vitro and in vivo, you mean?
Not between in vitro and in vivo, but this is also a good question, but I mean among individuals, so among different individuals.
Well, yes, there was high variability, of course, in results, even in the kind of symptoms you could see across animals. And it was clear, of course, in the in vivo experiment. Notably because during the in vivo experiment, we had animals that had maybe different ages. It was not easy actually to find a very homogeneous type of cohorts for the group. So and it may lead to quite high variability in the patterns or symptoms, but still we managed to have like quite similar patterns in terms of gene expression, upregulation, and regulation specific to each treatment, and they were all comparable to the in vitro experiment, but there was more variability in the in vivo than in vitro, as we would have probably expected.
Arnaud, sorry, can you please stop sharing your screen?
Of course, yes. It was in case
Okay.
there was questions. I would have to present
There is one question from Gaetano.
Well, first of all, thank you for the nice presentation. Actually, you got a very good data. And I have just a curiosity. I mean, if you change the host genetic background, do you expect the same results? I mean, you use a very, let's say, very selected breed of goats. If you change the genetic background of your goats, do you expect the same results or something
not
strongly changed?
It will depend on the breed. Of course, I mean, where the virus is more present, for example, it's endemic in Africa. If we are using some African breeds, for example, where they actually have, some level of resistance or tolerance to the virus, we will probably see something else. And also, the virus that is low virulence, the Ivory Coast 89, it's low virulent in this goat, but in some sheep breed, for example, it's highly virulent, and it's one of the interesting things, I mean, that we want to keep on explore first in vitro studies. It's really like compare the results in PBMCs in different in different breeds or different species. Of course, I mean, you cannot have, when you want to do transcriptomic, proteomic, you cannot have hundreds of samples because it just costs too much. So you want to start with a model that you understand well and for which you know what you expect, and that's why we went for this model.
Okay, thank you very much.
Thank you. There is another question.
Yeah, so I have a naive question regarding the interpretation of the result. So thank you for the very nice set of data that you present. I was wondering, because you spoke about the differential activation of the innate immune response between the strains, but what about the control of the innate immune response and the shift to the adaptive immune response? Because it seems that there is a delay and you have a drop in T lymphocyte afterwards that could be due to the balance of proliferation and apoptosis. And I was wondering whether finally there are not genes that are upregulated by the vaccine that would lead to better control of adaptive immune responses, and not this avalanche of activation.
Yeah, that's, I guess, one of the main key points where it will differ between strains is that for the vaccine strain or the low virulent strain, it will be able to enter the cell, interact with the host, but it will it seems to be more slowly, has less capacity to replicate. So it gives kind of maybe more time and capacity for the immune response to launch the full kind of response, then the innate and then the adaptive response. And in the case of the highly virulent strain, as those viruses are really very good at inhibiting the lymphocyte proliferation and really killing those cells, there you will really have a strong inhibition of T cells proliferation, and then a capacity to launch an efficient adaptive response, adaptive immunity response.
Thank you. Thank you, Arnold. We have to skip the discussion because for the coffee break. But I know that you are not available this afternoon, but I hope you are available this morning. If anybody has a question, take your opportunity to use the share, and I think Arnold will see your question and I hope we'll have time to answer it. I think we got a nice lesson on PPRV, but also got a nice lesson on what is possible. And I think Arnaud can also give you some advices on the methodology if you have some question on this. Thank you very much, Arnaud. That was a very nice story of VetBioNet TNA. Thank you very much.
Thank you very much.
And give applause, you will hear it. Thanks a lot. The next step is a coffee break, I think.
Oh, spero di farci. No, tranquilla, se no te lo date.
May I invite all the people outside to join us and also invite the people online to await a presentation by Professor Gaetano Donofrio from Italy. And he is quite experienced user in the infrastructure project. I think you already had a project in Nadir, if I remember correctly. Yes, and you had 2 projects within the VetBioNet with different topics. That shows that your interest is quite high in the infrastructure projects, and therefore I am really looking forward to your talk on the use of your bovine herpesvirus 4 based vaccines, which is a quite important question. Thank you for coming.
Okay, thank you very much for the organization, because I got this opportunity. Actually, I don't know anybody here, and probably you don't know me either, isn't it? But for this reason, it's been a great opportunity for me. So I didn't know anything about VetBioNet, to be honest. And then I got in touch with the Simon Graham in Pirbright Institute. We were doing something together, and I used to make these recombinant viruses. My main work is viral vector and gene therapy, let's say, okay, for animals and human beings as well. Now, I made this recombinant virus for Nipah virus and another one for PPRV. I said, well, what can I do with this virus now? I need to challenge some animals. I need to immunize animals. And they say, why you don't apply for TNA? I did it. Actually, it was very fast, very quick, okay, and very easily. Usually, if you apply for money for European community, it's a disaster. You know, you have to spend so much time writing, and you spend so much time. Here was very, very easy and fast. And this is actually great. Okay, we applied for 2 grants. One is assessment of immunogenicity of a BHE4 based vector delivering Nipah virus glycoprotein in swine. And the other one was PPRV ship vaccination with bovine herpesvirus 4 based vector delivering PPRV H antigen. Okay, we applied for these grants. They have been granted, both of them. They have been finalized. we published them, so everything went very smoothly. Now, before to speak about NIPA and PPFV, I want to give you a few information about the vector, okay, BHV4. Usually when you go to conference, you say, well, people ask you, what are you working on? And you say BHV4, they say BHV what? So they don't know what this virus is. Anyway, it's an herpes virus, okay? It's a bovine herpes virus 4. It's a gamma herpes virus. We don't know if it's pathogenic or not. Probably it's not pathogenic. I mean, when you isolate BHV4 from several lesions, okay, we try to isolate BHV4 from conjunctivitis, ulcerative mammary dermatitis, interdigital dermatitis, or vaginitis. Often you get out the virus from vaginitis. You make an experimental infection and you never get out the same lesion from where you got your virus. So doesn't respect at all the Koch postulate, okay? So we don't know if it's pathogenic or not. Actually, I believe that could have a residual pathogenicity, but not as a primary infection. You need something else together, okay? Now, is a gamma herpes virus like EBV or the other adenovirus, Kaposi sarcoma-associated herpes virus, ovine herpes virus 2, MHV68, and so on. The gene expression cascade is similar to the other herpes virus. You have immediate early, and late. The genome is quite big. It's double-strand DNA genome. It's 144 kb, okay? It's not easily to handle. Now, we made the first recombinant BHV4 by classical homologous recombination with using GFP. We put GFP inside of the virus, and we got a green virus. As you can see, you can infect several cell types coming from different animal species, and you could get cell transduction. Now, we tried before in mice. We injected the virus in the brain, and as you can see, we could have transduction of many areas of the brain, and the mice were fine. So wasn't pathogenic at all. And actually, we got an elongated chain of cells inside of the lateral ventricle of the brain, which were transduced by the virus. Now we try to characterize a little bit the cells, and they were let's say, a predifferentiation stage of oligodendrocytes. Next, now, we said, little or not pathogenicity, at least we don't see pathogenicity of this virus. Unlikely oncogenicity, we don't have any oncogenic virus inside the full genome. We don't find ORFs which can be associated to oncogenicity. Capability to accommodate large amount of foreign genetic materials. At least in our hands, we could stick inside 20 kb of foreign DNA, okay? Then capability to infect several cellular types coming from different animal species, and capability to maintain transgene expression during differentiation. We said it could be a good gene delivery method. That's why we decided to clone the entire genome as a bacterial artificial chromosome. The meaning is that to put the whole 144 kb in a single plasmid, okay? Now, we managed to get this bacterial artificial chromosome, so you can modify the genome inside is E. coli, and then get out the recombinant genome, introduce the genome in mammalian cells, and get out the recombinant virus, okay? So you can screen hundreds of recombinants in the same time in a single Petri dish, okay? So we made the bacterial artificial chromosome of this genome from several isolates from healthy animals. We isolated the virus from milk, for example, from mastitis, from interdigital dermatitis, from postpartum metritis. With all these, you can see there are a little bit different among them. Now, let's get in the project with the TNA, okay? The first one was assessment of the immunogenicity of bovine herpes virus sorbus vector for Nipah virus glycoprotein in swine. Everybody knows what Nipah virus is, okay? The reservoir is the bath. Through the urine can infect the swine. And in the swine, you can have amplification of the virus that can infect human being. And it is lethal for pig and human being as well. Now, we made 2 recombinant BH4 for 2 different glycoproteins, okay? Glycoprotein F and glycoprotein G. The first one is a fusion protein. We made the recombinant virus, as you can see. I don't know what the laser point is. Anyway, oh, okay. And as you can see here, the modification inside of the genome, you see that both viruses can express the transgene, okay? With a constituted virus. And as you can see, the virus bringing the glycoprotein is replicating similarly to the wild type virus. Now, the most important thing was to adapt the virus to swine cells. So we adapted the virus to swine cells, and then we grow the virus in the presence of swine serum. This was very important because when you make a vaccine for the vet field, the vaccine must be cheap, okay? So you have to reduce drastically all the methods because you need to reduce the cost of the vaccine. In that case, you have just to infect the cells in the presence of swine serum. You can harvest, okay, the soup, and you titer the virus, and you inject directly the soup with the virus, okay? Now, as you can see, we injected swines, okay? And is completely safe, okay? You don't have any detrimental effect to the animals. And then we start to make, actually this was mainly made in Pirbright, in the Simon Graham group. And as you can see, if you compare NIV-G with NIV-F and another virus, Alvac is a vaccinia virus, okay, expressing NIV-G. In terms of immune response, humoral immune response, you can see a very good amount of antibody produced by all of them, okay? This is a specific antigen for NIV-G and NIV-F. And you can see nice serum-neutralizing antibody, and you have cross-neutralization as well. Then we investigated the cellular immune response. As you can see, when you stimulate in a specific way with the full NIV virus, you have good immune response in terms of interferon gamma for NIV-G, NIV-F, okay? And actually the immune response from BHV4 NIV-G was much better respect to the immune response for NIV-G expressed by Alvac, okay? If you have any question, you can stop me and say, well, what are you saying, okay? Now, we finished this project and we published it because it was very interesting. Now we have a very big bottleneck. We could make the experimental infection, okay? You need a biosafety level 4 facility. I don't know if VetBioNet can help me for this issue. We got in touch someone in Germany, in Insel Riems. They have a very good facility there, but they seem to be very, very busy. So we are still waiting to try to make the experimental infection. Now, the other project was on PPRV. I'm not gonna spend time speaking about the virus, you know everything already. And we made a recombinant BHV4 expressing the hemagglutinin, the H of, and we made a preliminary study in mice, okay? And as you can see, we could get very good serum neutralizing antibody and a very good cellular immune response, okay? So we applied for money for TNA and we got the opportunity to make the second experiment in sheep, okay? We immunize sheep, a group of control with a BHV4, okay, without H, and another group just with PBS. The inject was, and we made the prime intramuscle, at 20 days, the booster, and at 42 days, the challenge, okay? Again, we adapted the recombinant virus to sheep cell serum from sheep. And then we analyzed the immune response before. As you can see, at day 41, you can have immune response, okay? And you can have serum neutralizing antibody as well. If you analyze the cellular immune response, again, we got a very good T cells immune response, okay? Now, you see that you have a better CD4 and CD8 and B cell as well, okay? Respect the unimmunized animals. And then we made the challenge. If you check the challenge, you see that you have complete protection of immunized sheep. And you don't have, this is the most important thing, you don't have shedding of the challenge virus, respect the virus to the control. Now, again, we managed to publish both study in mice and in sheep, and we've been very happy about this stuff. And now, as I told you before, I think that the study in sheep can be considered finished. We are just waiting if someone is interested to apply the recombinant virus on the field. And for the Nipah virus, we should do something. We should do something in a biosafety level 4. That's it. If you have any question, I'm very happy to answer you.
Thank you very much. There are questions.
Hi, thank you for your talk. I'm curious about the tropism, the cell tropism of the virus, epithelial cells, neurons, and also another question is I'm also surprised that there is no restriction of the host, that you can infect many hosts.
That's the nice thing about this virus. You can infect several cell types coming from different animal species and you can get infection. In some cell type, you have a full replication of the virus and CPE. In other cell types, you don't have any CP, but you can see the genome inside. I can show you something. No, you. Okay.
Can you go back to the presentation?
Doesn't matter. Okay, everything is dependent from the immediate early 2 gene. Immediate early 2 gene is a gene which produces a protein, which is a transcription factor that transactivates all the promoter present inside of the viral genome. Okay? Now, if the promoter of RTA is recognized by the pool of transcription factor of the cell host can be activated. Otherwise, it's not activated. But if you put your transgene with a housekeeping promoter, it can be expressed. So the secret is the pattern of penetration of the virus inside of the cells, which is quite peculiar for BH4 because it has few glycoprotein on the surface as GB, GH, and GL. That's it. So it can infect several cell types, okay? Epithelial, neuron, endothelial. If you see these other, non si muove. Let's see in goats. For example, this is was an experimental infection made in goats, okay? We tried to use this BHV4 with the luciferase and see what's happening, okay, after the injection. This is the site of injection under the tail, okay? And then you see that the luciferase is expressed, okay? And you can see that if you analyze the cell expressing the transgene, both epithelial and endothelial cells can express the transgene, okay? As you can see, these cells are going to die, these other cells, they survive. So, depends, okay? In some cells, immediate early 2 is activated, in other cells, is not activated. But this was a very important experiment. You know why? Because you see the expression of the transgene, you see the infection, but it's local, it's not spreading. So if you check for systemic infection, you will never find the virus. But virus is replicating in the site of inoculation. Did I answer to your question? Thank you.
Thank you. Are there more questions? We are a little bit running out of time, so we have to proceed. Thank you very much, Gaetano, for this very nice talk on this vaccine candidate. And in regards to your BSL-4 requirement, I think you have indeed to contact our colleagues at the FLI in Germany. They have this facility. There are not so many facilities. And maybe in Isidore, with the Nipah virus, there might also be some possibilities to look for Nipah virus as is an urgent zoonotic or pandemic question. Yeah, so keep in touch to Sasha and or David. They will may help you in proceeding with another infrastructure project. You're an experienced user. Okay. Then we come to the next presentation. That is a little bit different presentation as this will not concern a BSL-3 pathogen. It concerns a porcine pathogen, but the studies have to be done within high containment environment. And Jean-Claude will hopefully get us through his study and the results and the conclusions from the study. Jean-Claude, welcome to see you on the screen. Hoped to have seen you here in Tours. And I'm really looking forward to your talk.
Thank you, Norbert. You can hear me?
Yes.
Okay. So thank you very much for the invitation to present this work. So I will share my screen. It's okay. Can you see my screen?
Yes.
Okay. So this work was related to define if a respiratory delivery of an host-directed biological product could be used as an alternative strategy to antibacterial prevention of swine pleuropneumonia. So this work was conducted. Yeah, I was one of the PI involved, so I'm working in the Superstar field in the Center for Infection Immunity. So the idea of our lab is to develop alternative therapy to quench infection or avoid treatment failure in bacterial pneumonia. And by this, combine antibiotics, that is the standard of care in animals and in humans, with immunomodulating agents targeting the conducting airways. And also to characterize the respiratory immune defense that is involved in this process and how it is compartmentalized in order to define more, we say, more adapted immunomodulating agents. So our work is based on innate immunity and stimulation of innate immunity. And what is interesting in innate immunity, first, it's a built-in and universal system. It's independent of individual. It's conserved among most species, both farm and veterinary and humans. It's ubiquitous, so most tissue and body compartments can be activated. It's immediately inducible within minutes. And very important specificity is that immobilize multiple immune effectors and immune cells that have broad spectrum range of antibacterial activities and even antiviral activities. And this is less likely that bacteria develop resistance to such defenses. So it's a very interesting, with a natural system to defend our organism. It's a mechanism that is independent of antibiotic resistance and it's self-regulated. So we are interested mainly in the toll-like receptor, and especially the toll-like receptor 5. So these receptors are, most of them, surface exposed receptor. They can detect foreign, non-self antigen. Especially for toll-like receptor 5, the most famous is flagellin. It's a bacterial protein. And when flagellin is detected by TLR5, it induces a cascade of signaling that is feeding NF-kappa B pathway, making this transient activation. So what is flagellin? So flagellin, so let me see if I have a pointer here. So flagellin is a part of the flagella of bacteria, you know, this apparatus that is at the surface of bacteria that allow mobilities. It's composed of one structural protein that is called flagellin that is making this filament. Flagellin, here it's the flagellin of Salmonella enterica cerobartifimurium, but is most well known in terms of structure and activity. It's composed of 3 domains. Hypervariable domain D2, D3, that is what makes the flagellum serotype specific. And conserved domain that is composed of D1 and D0. So it's N and C terminus that come together to make this domain. And this domain is hyperconserved among bacteria. This D0, D1 domain are in fact the part of the molecule that is required for toll-like receptor 5 detection and signaling. So this is just to show you how it works. There is a specific motif in this region, in the D1 domain, that can be mutated to kill the activity. So can move to the next step. So how did I enter in the VetBioNet infrastructure? It was not really directly by VetBioNet, but more by another infrastructure that is called Transvac. 6 years ago, I worked with Transvac to analyze skin response to flagellin for a vaccination project. So in this project, we define different formulation, and yeah, we just summarize in 3 slides what we have done. So we injected different doses of flagellin. It was a native molecule from 0.1 to 10 micrograms, so in both sides of several pigs. And 4 hours after the injection, so it was the intradermal injection, we pick up by a biopsy of the skin, and we analyze this tissue using a microarray at this time and real-time PCR. So just to give you an idea of what we observed, it's if we analyze this gene SA2 expression in the skin compared to the buffer injected skin or a mock skin that was not injected, but were arbitrarily defined as a level of 1 of expression, we see that whatever the dose of flagellin we injected, 0.1 to 10 microgram, we observe a strong increase and significant increase of expression of the SA2 in the skin, meaning that something pro-inflammatory occurs in the skin. And using microarray, we're able to define several pathways that are very important in this signaling, especially the TREM1 signaling, the role of pattern recognition receptors, dendritic cell maturation that we were very happy about vaccination purposes, and also granulocyte adhesion and diapetesis. So meaning that flagellin is rapidly able to trigger a strong pro-inflammatory response in the skin. So coming back to the VetBioNet, so I was working mainly in the respiratory tract, trying to define how bacteria are dealing with the respiratory tract, how they hijack the immune system, and how flagellin could be used as an immunomodulating agent to prevent this infection by bacteria. So here it's just a schematic view of what we know about the signaling that was based on human cell and mouse experiment. So TLR5 is expressed by airway epithelial cells, several of them, and when it's triggered, it will induce both phagocyte-specific chemokine. This is very well defined, but we recruit neutrophil, monocyte, and other phagocytes, and also some antimicrobial molecules, especially beta-defensin 2 and catalycidin that were strongly upregulated. And we believe that this is contributing to protection against various infectious challenges. In between, at the beginning of the project, we are always already aware that the pig airway epithelium was able to respond to flagellin, and this was based on the work of Ignacio Caballero in RAE that show that airway administration of flagellin can regulate the response, pro-inflammatory response of epithelial cells, and in normal, I would say, and in some pathologic condition like cystic fibrosis. So this was a very important issue to start the study. So in this study that we conducted with the group of Wageningen and also with the help of Ignacio, the idea was to evaluate the effect of a single dose delivery of flagellin in pigs using a nebulizer. So it was an aerogen solo nebulizer, you see this device, and to define whether can trigger immune stimulatory activity in the respiratory tract. So using a drug and device system. And based on this, it was a go, no go for considering doing experiment to prevent infection in the pigs using Actinobacillus pleuropneumoniae. So what we have done is to design this experiment with VetBioNet consortium. It's to administer 3 groups of piglets with either buffer or flagellin, and on the flagellin moiety, we have 2 groups. One that was sampled at 3 to 4 hours. I probably in the next slide summarize that as 3 hours, and the group that was sampled at 24 hours. And we monitor different parameters in these animals. So injected 1.5 milligrams, so it's the administered dose, sorry, it's not injected, it's administered by the nebulization system. We expected to get 0.5 milligram in the animal. So very high dose to be sure that we are looking to something that is physiologically relevant. So just we based our calculation on this pioneer study that was conducted in pigs, especially with help of Ronan McLaughlin, that is an expert in aerogen device. So we know that using that, we can get 30% of a drug into a lung. So this is a setup that was done in Wageningen, thanks to Norbert teams. So as you can see, we the anesthetized pigs were connected to mask, and the mask were connected to this nebulizer. You can see here and here. And after the experiment, we sampled during the experiment we sampled different biological tissue, so the blood, the lung, the bronchus, the trachea, the nose, and also the bronchoalveolar lavage. This is a picture we set up to define the region of the lung that were sampled. So the first important observation that was done is that nebulization of flagyellin does not impact, at least in a significant way, the body temperature and the body weight. So meaning that it's safe to a certain extent. There is no major outcome due to the nebulization. We also observe that nebulization does not modulate in a severe ways blood cell content. Here we have monocyte, neutrophil, and eosinophil and other larger blood cell that were analyzed. We see that there are some changes, but they are, I would say, in a range that is physiological. Upon nebulization, we analyze different parameters. Here are the neutrophil content, percentage of neutrophil in two compartments, in the bronchoalveolar lavage and in the lung. And as you can see, there is an increase in the neutrophil compartment, both in the lavage, and this is significant, and in the lung, it's less significant, but there is a trend that is very, very strong. And it's what we expect based on other models that we have developed in the lab. The other important observation was that nebulization induces the production of cytokines in the bronchoalveolar lavage, but not in the blood, at least for these cytokines that were analyzed, so IL-10, IL-23, P40, and IL-12, and TNF-alpha. As you can see, the stimulation is associated to cytokine production. That is something that was expected. We next use the tissue to make some omic analysis. So this is a first preliminary experiment of real-time qPCR on lung, bronchus, trachea, and nose tissue. So here we show you a few genes that we analyzed before going to the transcriptomic, more deep transcriptomic analysis. As you can see, IL-1 beta is upregulated, and yes, more or less significantly, but it's upregulated in the different animals. So for 4 animals per group. It's the same for CCL2. You can see that there is an increase. And also for PGLY, PR2, that is a peptidoglycan associated receptor. To get a more complete picture of the impact of flagellin nebulized in the tissue, what we have done is to conduct a total tissue RNA-seq analysis. So here it's the analysis we conducted on blood, lung, and the bronchus tissue. As you can see, there are plenty of genes that are regulated over time. So from 0 to 24 hours, about 2,000 per condition. And this is a pattern of a gene. So the genes that are upregulated and stable over time, but one that are upregulated 24 hours, and the one that are just upregulated or downregulated at 3 hours. We conducted a first analysis of genset enrichment analysis, a GSEA, And we find in bronchus only a very strong picture, a pattern, sorry, of immune regulation with all the immune response processes that are upregulated, and what is also related to response to external stimuli and to leukocyte infiltration. So this is really also something that we are expecting to a certain extent. For a moment, we have observed that in bronchus. With GSAR, we are conducting over-representation analysis to define if we can further define pathway in other piece part of the tissue. So just as a conclusion of this study, the main conclusion is nebulization of flagellin-induced immunostimulation in respiratory tract of pigs. So we achieve, I would say, the first of our objective, which is based on granulocyte presence in blood and lung, a significant increase of expression of pro-inflammatory gene. We are also happy that there was no sign, I would say, of severe toxicity because of the hematological parameters, sorry, the white blood cell count, the clinical signs were pretty in a moderate way modified. And also what I have not shown here, we have also analyzed the histology, and again, based on the histology scoring, we are not, it was not associated to major pathology or a severity of a treatment. So what are the next step? So we're still processing for RNA-seq some samples because we did that in two phases. So we are currently analyzing the nose and the trachea, and also the different biopsy of the lung, because we just analyzed one piece of the lung, but now we are analyzing the rest of the samples. We'll expand the omic analysis of these samples, and we are also interested in making some cross-species analysis. We have also data from the mouse tissue and also from non-human primate that would like to see if it's what are the commonalities and the specificity. And the second objective, as I mentioned initially in the talk, was to make an efficacy study using Actinobacillus pleuropneumoniae that gives some this pneumonia, this pleuropneumonia. And we started that in VetBioNet in BSL2 conditions. So we set up the infection, and thanks to Norbert team. And this was continued with a group of Ignacio at INRAE with a national funding, IGET, ANR funding. So just to give you some information on what has been done, but I will not give results. I will just let you know what has been done. So the idea was to infect the animal with Actinobacillus, so intranasal infection, and then we split the animal in three groups. One group that will receive only buffer that was nebulized, that is the control of infection. One that will receive flagellin that is nebulized and subtherapeutic antibiotic treatment, and one that receives subtherapeutic antibiotic treatment. The idea is to see the superiority of the treatment with flagellin on top of the antibiotic treatment. That's why we use subtherapeutic antibiotic. And what we have done in this experiment to analyze the clinical signs, the biology also, the cell counts and cytokine, bacteriological evaluation of the system, and also immunological evaluation. And we are conducting omics analysis on this on some of the tissue sample that we have obtained from the animal. So this is ongoing, and I think that Ignacio and Norbert will present that soon in a conference. So I would like to thank the people that were involved in this work, especially Mara Baldry, that is a colleague of mine, and you will see how she contributed in the next slide. Norbert Stockhofer, because I started to work with him on Transvac, and when I wanted to develop this challenge study in the pig, it was very helpful to have him on board because I already discussed a lot with him during the Transvac project, and it was a way to also enter in the VetBioNet infrastructure. And Ignacio, that I was working with him before the VetBioNet project, using through Vaincre la mucoviscidose, the Cystic Fibrosis Foundation in France, on flagellin signaling, and now we get this national funding, VetBioNet, and we are still continuing collaboration, but very very exciting. And also I wanted to thank in this acknowledgement slide Professor Stephen Gordon, but from University College of Dublin, that makes some experiment with nanostring to validate our RNA-seq data. So it's a very important collaborator also. So in the next, I think 2 slides, just what I want to say, it's this project was a leverage for, we say, for the next steps. So it contributes to the development of a project that I'm coordinating at the moment that aims to use this flagellin in a phase 1 clinical trial in humans. So we expect to start that next year in 2024. And the data that we have generated during VetBioNet will be very strong and consolidating, I would say, the whole picture that we have on the possible use of flagellin in your treatment. So these are the collaborators that are involved in this project. And the second, we say, adding value and on this the use of a VetBioNet, access of a VetBioNet infrastructure, it's yes to consolidate the proof of concept for immunotherapy of infection. Also to perhaps a way to go back to the respiratory vaccination in addition to immunotherapy of infection. And I have to say it was also very important way to build outstanding collaboration with the veterinary field, and I was very happy to interact with Norbert and Ignacio for this study, and it was really we say cross-fertilizing project. So I want really to thank warmly the collaborator and VetBioNet for the funding of this project. And I'm done. Thank you.
Jean-Claude, thank you very much for this nice presentation. And I have to confirm that nanostring analysis are very important and have to be finalized to be sure, to be honest. But have been done in the joint and research activities within this. So we have a cross link between the international activities and the joint research activities, which also shows the different lines which you can follow within such a project. But I'm really impressed also on your how you proceed with this project. You started already more than 10 years ago, 15 years ago, I think, and you're still looking for possibilities in the research field to look for collaborations. Are there any questions for Jean-Claude?
Thanks, Norbert. Stephen Anderson from the Morden Research Institute. Thanks for your talk. That's really interesting. My question's around the nebulizing of these pigs. Have you tried ever doing that with them conscious? I see that you just had two on the table under anesthetic. I'm just wondering about the variation in, you know, obviously if you're just doing 2 at a time, if you're doing a big group study, you're going to get quite a lot of variation, you know, just inevitably by doing them 2 at a time. So I just wondered if you've tried any conscious nebulization, because that may give increased, certainly we found with calves, doing them conscious does mean they're breathing a bit deeper, you're getting a bit better penetration into the lung than you do under anesthetic. So just wondered if that had been looked at all. Thank you.
No, we have not done that. I think for the toxicology study that we will conduct soon, it will be done in some animals. it requires some specific, we say, device, because you need a plenum chamber to have a, we say, homogenized aerosol for all animals if you want to, to nebulize 5, 6, 7 animals at the same time. So it makes the things a little bit complex, I would say. And yes, for other animals, I'm not sure it's very easy to maintain them under this with a mask without specific constraints, especially if they are conscious. So I don't know. It's I'm not expert in the field. I'm sorry, but I think if you want to have something that is very homogeneous, you need to have a specific system to deliver the aerosol to be sure that it's properly delivered.
Maybe I can add on this. We did this, in fact, in anesthetized animals, and because we are expecting that the animals do not like this mouse or direct inhalation. We will have soon a study where we start training pigs to accept the mouse, but I expect that it takes at least 1 week to train them by continuous or by regular and high frequency exposition to this system. But piglets can be trained. I'm pretty sure that they can be, but it's quite time and work consuming. The other thing is that you would like to see, and I personally also would like to see, that you have the possibility to have groups inhaling the material at the same time in a conscious, but then you have a whole body exposure, which means that your control of exposition gets much less. So this is a complex thing, problem. We will discuss it during a workshop we will have at the end of March on aerosol administration. You know about it, and I'm really lucky if you, I'm really would happy if you would raise the questions there also. Thank you very much. Any questions more? If that is not the case, Jean-Claude, very much for your presentation.
Thank you.
And I'm really looking forward to finalize this study. And hopefully for some possibilities to proceed with this interesting concept of using the innate immune response to fight infections. Thank you very much. We're coming to the next presentation. This is Caroline Denisvet. No. What is it? Ah, Dolores, yes, of course. Sorry, I made a mistake. Yeah, I expected Dolores to be here, but on Sunday we were informed that she will not make it due to the strike, but she was so kind to join online, and I see her in the screen. Dolores, welcome to the meeting. It's really a pity that you're not here. We would have liked to talk to you, and I'm happy to your presentation on African swine fever study you did together with the group from, it was APHA, I think, huh? Yes, yeah. And the floor is yours.
All right, thank you very much. I'll share my screen. Let me see. Okay, can you see the slides? Can you hear me?
Yeah, very well.
Okay, can you see my slides?
Yes, the slides are
great.
Beautiful.
Thank you very much. Yeah, really pity I cannot be there with you, but I'm from here, from Sweden then, freezing and thinking of you in the warmth there and enjoying being together. I will present this very interesting project on the comparative evaluation of the pathogenetic mechanisms and immune response in wild boar and domestic pigs experimentally infected with an African swine fever virus of genotype 2, which is a strain circulating in Europe. I would like to start by acknowledging the collaboration that we have had through this project. We in Sweden do not have any BSL-3 containment facilities for large animal experiments, so we were extremely grateful to VetBioNet and to our collaborators for this opportunity then to conduct this study. This study was done by a collaboration between Animal and Plant Health Agency in the UK. And here we had a pathologist with a long and excellent experience on African swine fever, Pedro Sanchez Cordon, and his team. And Pedro was really a big enthusiast, and we have learned a lot from him. So I'm very grateful, especially to Pedro. Afterwards, he moved to Spain, to CISA, but the collaboration continues. We work together also with the Pirbright Institute and with ANSYS, and all of us met several times through the course of the Cost Action ASF Stop. So we knew each other. We knew, of course, Weybridge,